I'm about to break it down, break it down. Okay, so for blood, remember what all of the cell types are and what they do. Remember what plasma is and what we find in it. Okay, so cells, form and function. What do they look like for lab practical? What do they do? Processes to worry about from blood. Erythropoiesis, how we get our red blood cells. The life cycle of a red blood cell. Hemostasis, all of those things are important. What else for blood? Make sure you're comfortable with the components, like plasma, what's in it, and what function does it serve. Then for the heart, what do you need to know? The anatomy, all the anatomy. There's lots of that, already covered in other videos. As far as physiological things to be able to describe or list or understand, I would say conduction cells and the behavior, or our pacemaker cells and the conduction of impulses through the heart's conduction system. So the heart sets its own pace, understand how that's happening through this conduction system, and then understand our contractile cells. Understand the cardiac cycle. Understand the things that affect cardiac output. What else? From the heart. See, I always get nervous about doing a video like this because if I leave something out and I test you on it, you could get mad at me. So that's not going to happen. Everything that has been covered in a lecture or a lab video is testable. And everything in the book that relates to that is also testable. So heart, anatomy, physiology. Yeah, I think we're good on that. And then for blood vessels, again, anatomy and physiology. What are the different layers look like in each of our different vessel types? What are they serving? How is blood pressure controlled? It's going to bring a whole lot of things together. So for our blood vessels, what are things that you need to be aware of from that chapter? So know about the structure of the blood vessel wall. Know about our different types of arteries. Elastic, muscular, arterials. Know about our different kinds of capillary beds. So know about our different types of veins, know about blood flowing through them, so as far as physiological processes go for that, know about blood flow, know about blood pressure and the things that affect it to the level that we talked about in class, know circulatory routes as far as they apply to what you need to know in your lab manual. And that's kind of like an overarching idea of everything that you need to be comfortable with. You can also look at your exam review documents that are available. So yeah, have fun with that. See you soon.
Hello, welcome to my coronavirus classroom. My name's Janessa Jacobs, and this is the Cardiovascular System Exam Review. I'm going to try and do this without editing, so we're going to jump right in. I already know that I made a mistake because I'm tired and it's late at night. So in this first question, let's just look at it, we'll talk about it, and then we're going to race right to the second question. Blank cells are formed elements found circulating in the blood. A, red blood. B, C, megakaryocytic, or D, all of the above. Blank cells are formed elements found circulating in the blood. All right, well, the correct answer is not up here because the correct answer is red blood cells and B cells are formed elements found circulating in the blood. So the correct answer should be down here, E, A and B only. Because megakaryocytic cells are the cells that hang out in the red bone marrow and stick out those little feet that get sheared off as platelets, the other formed elements. So if this question instead had platelets here and said blank cells are formed elements found circulating in the blood, the answer would still be E because platelets aren't cells. So just be aware of that. Now we're going to race to the next question. And what purpose does the coronary circuit serve? A, it delivers oxygenated blood to the myocardium. B, it drains deoxygenated blood from the heart. C, it acts as a continuous source of nutrient and gas exchange. D, all of the above. Yeah, the answer is all of the above. So cardiac muscle has to use aerobic respiration. It relies solely on aerobic respiration. Skeletal muscle can use anaerobic respiration, so it can do different stuff than the heart. But because the heart cannot, then we have to have a continuous supply of nutrient and gas exchange. So we always have glucose and oxygen available for aerobic respiration in cardiac tissue. Okay, true or false? All arteries carry oxygenated blood. The answer is false. Why is that? Because all arteries carry blood away from the heart, right? And oxygenated blood has to be carried away from the heart to the circuit, and deoxygenated blood has to be carried away from the heart to the lungs. So that's false. True or false? All systemic arteries carry oxygenated blood. This is what is true, and when most people think of arteries, this is what they're thinking of, is the systemic arteries. And that's because there are a bunch more systemic arteries, and that's because your entire system is a lot bigger than the pulmonary system. Doesn't mean that your pulmonary arteries aren't arteries carrying blood away from the heart. They are, it's just deoxygenated blood. When I first wrote this question, because I was tired, I actually wrote all blood vessels carry oxygenated blood. And is that true or false? Well, of course that's false, because our veins in the systemic circuit carry deoxygenated blood, and our arteries in the pulmonary circuit carry deoxygenated blood. I need to go to sleep. Okay. Precapillary sphincters can or will A, control blood flow into the capillaries, B, cause blood to flow directly from the meta-arterial into the postcapillary venule, C, open when tissues need nutrients, D, all of the above. The answer is D, all of the above. Okay, and why is this? So precapillary sphincters, remember, if they're constricted, then blood is going to go directly from the meta-arterial to the thoroughfare channel. And why would that happen? Because we are good, we don't need nutrient and gas exchange. So if we do need nutrient and gas exchange, then we would open these. And what happens then when we open those is that we are exerting control of the blood flow into the capillaries. Oh, but what if we don't need nutrient and gas exchange? Then we can be constricted and cause blood to flow directly from the meta-arterial into the postcapillary venule. Okay, blank cells are phagocytic. A, basophils, B, macrophages, C, reticular cells, D, all of the above. Yeah, the answer is B, macrophages. What do basophils do? They release our inflammatory chemicals. Basophils release inflammatory chemicals. Reticular cells are the supporting cells that secrete reticular connective tissue in lymphatic organs. Okay. Cells in the heart's conduction system. A, require nervous system stimulation. B, do not require the same ions as other conductive cells. C, set the pace of the heart. D, all of the above. The answer is C, they set the pace of the heart. So why don't these other answers work? So they do not require nervous system stimulation because they set their own pace. The conductive cells do that because of those pacemaker channels, the slow sodium channels that allow sodium to trickle in the cells from their resting membrane potential of -60 millivolts to the threshold for those voltage-gated calcium channels, -40 millivolts. So that's how they're setting the pace. The nervous system, the autonomic nervous system, can get involved if need be to increase and then decrease the heart rate, but the cells in the heart are setting the pace of the heart, and they do require the same ions as other conductive cells. The other conductive cells that we have would be the cells that can conduct action potentials. So what can conduct action potentials? Neurons do it down their axons, and muscle cells can conduct action potentials along their plasma membranes. So they all are using the same ions. So yes, they require the same ions, they're just coming in through different channels. And I am fading fast, so you're going to review which different channels they are. Okay, the most important mechanism of capillary exchange is A, diffusion, B, osmosis, C, filtration, or D, all of the above. Yeah, the answer is A, diffusion, because diffusion is the most important for or it's how glucose and oxygen are moving. So, and tissues need that the most. So sorry if I went over that answer too fast. Again, I'm getting really tired. So if you want to, when I read the question, just hit pause while you think about it. The vitamins you take every day are floating around in your blank. A, blood plasma. B, red blood cells. C, buffy coat. D, all of the above. Okay, they are floating around in your blood plasma. So everything that is not a formed element is floating around in your blood plasma. One of the things that I really don't like is when I say something like, true or false, red blood cells carry nutrients. And I hear, true. No, they do not. Red blood cells carry respiratory gases, oxygen and CO2. Nutrients are things like glucose, amino acids, vitamins, and minerals, and they are not carried on red blood cells. Those are floating around in your blood plasma. So the vitamins that you take in the morning, or you should be taking in the morning, are floating around in your blood plasma. Okay, the interventricular septum. A is between the ventricles. B contains all of the heart's conduction system. C contains only cardiac muscle tissue. D, all of the above. Yeah, the only correct answer here is A, is between the ventricles. And that's because the heart's conduction system starts at the SA node, which is up in the atrium, continues in the AV node, which is up in the atrium, and then continues through the interventricular septum. And it also, this answer doesn't work because the heart's conduction system is insulated by that fibrous skeleton of the heart, so that as we're passing through the interventricular septum, that impulse isn't stimulating myocardiocytes. Because if it did, then we wouldn't be able to contract from the apex upward, and that's what we have to do. Okay, the steps of hemostasis are A, vascular spasm, B, platelet plug formation, C, coagulation, D, all of the above. Wahahaha! What are the steps? I didn't blue out our answer, but the answer is D, all of the above. So review those steps if you need to. The pericardium is A, the serous membrane of the heart, B, unique among serous membranes, C, comprised of multiple tissue types. Composed, comprised. D, all of the above. All of the above. So it is the serous membrane of the heart. It's unique among serous membranes because it's got fibrous connective tissue on the outside. In the fibrous pericardium, it's that dense connective tissue. And that would make it multiple tissue types because the inner type, which is what normal serous membranes have, is just our two different layers of simple squamous epithelial tissue. Simple squamous epithelial tissue. It is more important to fix blood pressure when it is blank than when it is blank. A, it is more important to fix blood pressure when it is high than when it is low. Or B, it is more important to fix blood pressure when it is low than when it is high. Yeah, the answer is B. If you have low blood pressure, you're going to lose blood flow. And that's no good. You lose blood flow, you die. So that is much more important. That's why we have so many pathways in the body to correct blood pressure. So make sure you review all of that before the exam. And hopefully Chip can get you a good, nice negative feedback loop comic up this weekend or something to review it. Okay, what else? That's it. All right, I have to say goodnight. I'll see you soon.